the topic of discussion for the last 2, 3 years. Every oncology conference meeting that we go, people talk about CAR T cell therapy. And it is very interesting to note why we are all excited and what this kind of treatment means for us. So we are of course going to talk about CAR T cell in the context of blood cancers. And as of now, it's only blood cancers which are where CAR T is approved for treatment. But we are hoping that this will be used for many different kinds of cancers, including various solid tumors. So since we are going to focus on blood cancers, and this is a hematology, certificate course, let's see how common are these blood cancers, right? We are talking of various kinds of treatments, including last time we spoke about bone marrow transplant, but how common are cancers, blood cancers? And I use this acronym called LLM, leukemia, lymphoma, myeloma, because these are the three major kinds of blood cancers. And if you look at the number of new patients who will be diagnosed in a year, then this is the population statistic. This is, what you would find in most of the places. Of course, around the world, the incidence varies a little bit. Partly it is because of the data collection mechanism. So what that means is in India, we are able to, we have a national registry, cancer registry, which ultimately reports to the international registry managed by WHO, IR. Dr. Tina Matthew, I think this, by chance, things are being written on the screen by you. Could you please erase that, Dr. Tina Matthew? Yeah. So, we collect data from different parts of the world. Oncologists do that. There are cancer registries, but from India, we probably collect data of less than 10% of the cancer patients. So you see, 90% of the patients, we don't have data. And this brings in a kind of bias called as sample bias. And thus, depending on how properly the data is collected, you will have slight differences in the number of new patients per 100,000 or million population per year from different parts of the world. But broadly, new blood cancers patients are 40 per lakh population per year. So if you extrapolate this to our country, we are seeing 500,000, 5,60,000 patients are theoretically diagnosed. Again, we don't have the real data, which means per day in our country, we have about 1,500 patients getting diagnosed. So that truly is a lot, right? And hence, maybe it's important to understand how we treat. And we have gone through this earlier, but I would like to summarize in one slide. Usually, what we do is cancer cells are rapidly dividing cells. They divide very rapidly. They multiply. They also die rapidly, but on the whole, the number of cells increase. So what we do is we give certain chemicals. These chemicals, whether you give them subcute, IV, intramuscular, oral, ultimately the chemicals go in the blood. And since the blood goes all around the body, this chemical is spread to all parts of the body. And every cell which is rapidly dividing will eat up this chemical. And since this chemical is a toxin, it will kill the cell. So chemotherapy is essentially a toxic chemical which is eaten up by rapidly dividing cells, which are, supposed to be cancer cells, but it will also be eaten up by other rapidly dividing cells. So hair follicle, the gastrointestinal, epithelium, so mucositis, diarrhea, hair loss, all of these are side effects of chemotherapy, because these normal non-cancerous cells will also die because they are rapidly dividing. They will eat up the poison, the toxin, the chemical, and that chemical will kill them. So that's chemotherapy. That's a nutshell, right? So what it essentially means is if you have a village which has, say, 500 terrorists, okay? Or if you have a city like Bangalore, which has 500 terrorists. And what military does is they send a plane and they drop hundreds of bombs. Now, these hundreds of bombs will kill the cancer cells, but can also kill normal citizens. That's exactly what chemotherapy does. It's just blind bombing. It will kill any cell which is rapidly dividing. But of course, nobody likes blind bombing. Everybody will say, no, no, why is military dumping bombs on Bangalore? Why don't you instead send people with a gun and only kill those targets which are actually terrorists, right? So targeted therapy. The police and the military should not be randomly bombing villages and cities, right? Target the terrorists. Similarly, in oncology, we want to target the cancerous cell so that only that cell dies, and there are minimum side effects on other types of cells. That's targeted therapy. And we have quite a few targeted therapies available now. Of course, starting from imatinib, which targets the Philadelphia chromosome. So Philadelphia chromosome, that is BCR-ABL mutation, causes CML. So you give a targeted therapy, only the CML cells will die. That is chronic myeloid leukemia, right? And that's targeted therapy. It's very different from chemotherapy. It's very simple. It only works on the bad cells and there are no side effects, right? Then somebody says, but what about boosting immunity? Why don't you use something to boost the patient's immunity? Well, you're right. That's a very valid question because the core concept, the core concept of cancer, okay, is that one cell goes bad. One cell, the DNA, the chromosomes, the software in the cell gets corrupted, right? And that corrupted software will make the cell multiply unnecessarily and spread and damage the body. Yeah. So it's a DNA defect, it's a mutation. And that's one part, right? So you say that, yeah, that guy was so good. And you see, I never knew he was a terrorist or he became a terrorist later. That's because a good person's brain, the software in his brain was corrupted by someone, and now that person has gone from being a nice young boy to a terrorist. Similarly, a normal cell can corrupt to become a cancer cell, right? So that's the first part. The second part is immunity. So you can say, yeah, of course, that guy was corrupted by someone, he became a terrorist, but what were the police doing? Why didn't the police catch him? Had they caught him early, then we would have neutralized that guy or put him in jail or whatever. But because the police failed, because the immunity failed, that one terrorist becomes two, becomes four, 800, 200, 500, right? Similarly, when a cell becomes cancerous, our immune system should have identified that this is not my cell. This is a cancer cell and I need to kill it. And trust me, your immunity and my immunity does this all the time without knowing. Without knowing, our native immunity keeps on killing bad cells. It keeps on killing autoimmune cells. It keeps on killing cancer cells. It keeps on killing infected cells, right? So immunity is supposed to be killing the abnormal cells, typically the cancer cells. But if the cancer cells are growing, means your immunity has failed, your police failed. So oncogenesis, conceptually, how a cancer develops is one cell goes bad and the immunity also fails to control it, kill it early, right? Both the things have to go hand in hand. So everybody says, no, 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 what about it? Why don't you use immunotherapy? And sure, we have been using immunotherapy. The first medicine was something called as rituximab. Rituximab. What rituximab does is it is essentially antibody. You create antibodies against one protein, a protein which is there on cancer cell. Okay, a cancer cell has a protein, say CD20, CD20. You create antibody, you create immunoglobulin against it, right? And then you take that immunoglobulin and you give it to a patient who has lymphoma, because some lymphomas have CD20. that immunoglobulin will go and get attached to the cell which has CD20 protein, and then the body's immunity will kill that cancer cell. Okay, so the immunoglobulin goes and gets attached to the cancer cell, and then rest of our immunity will understand, okay, this is attached, means this must be a cancer cell, I have to kill it. So that's immunotherapy. Now we have many more drugs. So blinatumomab, etc. There are drugs against CD19, CD22, etc. And then last week we saw that if blood cancers can't be created, cured with some patients with chemotherapy and immunotherapy, then we do a stem cell transplant and we change the immunity. We change the immunity so that the new immunity may be able to kill the cancer cells if they start growing again. So broadly, these are the four methods for treating blood cancer. Blood cancer. Of course, in solid tumors such as breast cancer, colon, we have surgery and radiation also. That's a different thing. We are focusing on blood cancers with chemo, targeted immunotherapy, and stem cell transplant. But of course, our current treatment is not perfect. It's not perfect. It's not perfect because we can't cure everyone. There are some patients who don't get cured. There are some patients in whom there is initial control, but the cancer comes back. There is a relapse, right? So they may be resistant to whatever you have used, or you want to control the cancer nicely before doing a transplant. Yeah. And you're not able to do it with certain chemoimmunotherapy drugs in some patients. Some patients, they work beautifully, some patients, they don't work. And of course, like I said, there is toxicity of chemotherapy. So if in an elderly patient or somebody who already has a cardiac problem, renal problem, etc., they are not able to tolerate the current treatments available for blood cancer. And hence, it's the treatment which might actually kill the patient, right? The side effects which may kill the patient. Now, luckily for us, most of the treatments that we use today, they do not kill patients, because over 50, 60, 70 years of cancer treatment, we've realized which kind of medicines we should combine to form a protocol. What dose should we give? What dose we should not give? How do we manage the side effects? So today, the number of patients who are dying because of cancer treatment are probably less than 10%, right? 90% of the deaths are because of uncontrolled cancer more than anything else, right? But still, even 10% is not good enough. So now let us see how this new kind of treatment, CAR T cell therapy, how does that work, and how does this treatment overcome the shortcomings of our current treatment? Yeah, like I said, there are two major concepts of cancer. A normal cell becomes cancerous and the immunity fails to kill it. How can the immunity fail to kill the cancer cell? How can it fail? Okay. So if we ask that, okay, when there were only one or two terrorists, how come the police didn't kill them, catch them, put them in jail? The police will say they looked like normal people here. They looked like normal people. They were going to work, they were going to college. They were not visible to us. They could not differentiate normal from terrorists, normal cell from cancer cell. Okay, how do we differentiate? So the cancer cells can often become less visible to immune system. The terrorist will ensure that he does not seem threatening. He's not going to walk around with a gun, correct? He's going to try to be a nice person, and so that he is not visible to the police and the neighbors. So the cells around the cancer cell, the normal cell surrounding the cancer cell, just like the normal neighbors who surround the terrorist will be very nice to them, so that they don't realize he's a terrorist. Others, they will call the police. So many times we hear the neighbors will say, hey, we never realized he was a terrorist. He was staying in our apartment for 6 months, right? So similarly, cancer cell can silence the neighboring cells so that there is no alert, so that the immune system is not activated. This is called as immune evasion by malignancies. And almost every cancer, in every cancer we see this, right? What else? I told you, we've been using immunotherapy for some time. We've been using antibodies, interferons, blah, blah, blah. And now we have CAR T cell. Now let us see what is this CAR T cell, okay? So in CAR T cell, what we are actually doing is we are making immune cells smarter. We are making immune cells smarter. What that means is you had a police and you had the military, and they were not able to identify the terrorists. So the terrorists multiplied and caused havoc. Now you should train these guys so that they can identify the terrorists properly, right? So you take normal army people and you train them and you make them black cat commandos. You make them black cat commandos who are very good at identifying and neutralizing terrorists. Similarly, you take a patient's T lymphocytes, T lymphocytes, and the patient's own T lymphocytes were not able to identify the cancer. So you make these T lymphocytes smarter, smarter, so that they can identify the cancer cell and kill it. It's exactly like that. You make the immune cells smarter. So you take a T cell, CAR T, T cell, and make that T cell smarter. How do you make them smarter? Okay. Now, this is fascinating. I find this amazing. And if you look at this picture, this is a normal, healthy cell, normal cell. This is a cancer cell. This cancer cell has an antigen at the cell surface. For example, CD19. So let's say this is a B cell lymphoma, B for Bombay, B cell lymphoma or a B cell leukemia, ALL, acute lymphoblastic leukemia. Now, B cell lymphoma and leukemia have this CD19 protein. It's an antigen. Now, the patient's own T cell is not able to identify it, not able to kill it. So you make this T cell and convert it into a CAR T cell and make it smarter so that now this CAR T cell can identify CD19, attach to it, and kill the cancer cell. Okay, that's the core idea. So, first of all, you have to figure out which protein is there on this cancer cell which is present, and it should not be present on other cells. Otherwise, our CAR T cell will also unnecessarily kill normal cells. And this protein should be essential. So when you get attached to this, when our CAR T cell get attached to this, it should be able to kill the cancer cell. It should be essential for the cancer cell. It should not be seen on normal cells, right? And if we can do this training to T cells and convert them to CAR T cell, maybe they can identify and kill the cancer cell, right? That's the fundamental hypothesis. That's the fundamental concept. How do we do this? How? How do we do this, right? How do we achieve this? So effectively, what we do is we figure out how does the antigen antibody reaction happen? How does it happen normally? And why is my normal T lymphocyte not able to identify? Why? What's the problem? Okay. So what we do is we figure out the normal antigen antibody reaction. We figure out the proteins which are required in this. Now, when you know the protein, you can find out the gene because this protein is on the cell surface, right? Then there is cytoplasm of the T lymphocyte, and then there is a nucleus. In the nucleus is the gene. There is the gene for the receptor for CD19. This is called as T cell receptor. T cell receptor, TCR. So you know that there is a T cell receptor which can attach to CD19. Once you know there is a T cell receptor protein, you can look at the DNA of the T lymphocyte and say, okay, now I have found the gene of the T cell receptor. That gene is the DNA, the software code. Okay. So you get the genetic sequence of the T cell receptor which can attach to CD19. And you now want to modify this. You want to modify this DNA sequence so that it is able to identify the cancer cell and kill it. Okay. So you modify it, and that is called as genetic engineering. In genetic engineering, what is done? You see, this is a normal T cell receptor. They add multiple other things to the T cell receptor to make the cell smarter, to make the cell more effective. So you modify the gene of the T cell receptor. Yeah. Now this modified gene, I want to put it into the patient's lymphocyte. I want to put this modified gene into the patient's lymphocyte so that the patient's lymphocyte can then make a modified protein, a modified T cell receptor. I want the patient's T cell lymphocyte to make the smarter T cell receptor, modified. But how do I put this modified gene inside the cell? How do I do it? I can use a virus. So we know that there are certain strains of lentivirus, adenovirus, retrovirus. These viruses, okay, there are strains available today where these viruses do not harm humans. Okay, so scientists have done a lot of research in the last 30, 40 years, and they've identified pathogenic viruses, non-pathogenic viruses. In the non-pathogenic viruses, we have to figure out, we have to get a virus which can enter the lymphocyte and not harm it. It should enter the lymphocyte and not harm it. So you get a non-pathogenic, safe lentivirus. You put the modified DNA inside the virus. We collect the patient's lymphocytes using apheresis. You remember I spoke about apheresis in the bone marrow transplant lecture. So, there is no surgery. The patient is lying on the couch watching Netflix. There's one needle in this hand, another needle in this hand. Little bit of the patient's blood goes in the apheresis machine. And the apheresis machine will centrifuge the blood and we get lymphocytes. So those lymphocytes are patient lymphocytes are collected in a bag and they are sent to the genetic engineering lab. The patient's lymphocytes are sent to the genetic engineering lab. Then they take the lymphocytes, they put them in a dish, a petri dish with culture medium. That means the lymphocytes need food to stay alive. The lymphocytes need food to grow and multiply. So put them in a petri dish with nutrients, with culture medium. Okay. Now these lymphocytes are the patient's lymphocytes who were not able to identify cancer cell. They were not able to identify CD19. Now in these, I will put I will infect the cells with lentivirus, which has a modified DNA of the receptor. These viruses will go inside the T lymphocytes and infect them. When they infect, what it actually does is this DNA then goes into the lymphocytes DNA. That is called as transduction. Transduction. That means the modified DNA of this receptor will come out of this virus and enter the lymphocyte's nucleus. Transduction. Okay. So now this lymphocyte has modified DNA. And now that lymphocyte's nucleus will turn that DNA into RNA and RNA into protein. And that protein will come out and stick on the surface of the lymphocyte. That protein will come and stick on the surface of the T lymphocyte. And then it can identify CD19, catch a cancer cell and kill it. Okay. So we have modified the CD19 receptor of the lymphocyte by passing inside a modified DNA, modified gene using lentivirus as a vector. The lentivirus is a vector, which means that the lentivirus is just a package. It's just a package. It is not harming. It is just a package. It's like a parcel, a courier, which takes the altered DNA. Okay. Now, this altered DNA has some of the original receptor, some new sequence. And because it is a combination of original and modified, it is called chimera. Chimera. In Greek mythology, there is an animal called chimera. Chimera has the head of a lion, or rather, body of a lion, and head of one animal and wings of another animal. It has different body parts from different animals. In our culture, in our mythology, it is similar to Lord Ganesha. Lord Ganesha has the body of a human being and head of an elephant. So that's a chimera. Okay. Now the antigen receptor, this CD19 antigen receptor is a chimera. Hence, it is chimeric antigen receptor, CAR, chimeric antigen receptor T cell. So you have changed the T cell receptor and made it chimeric by adding different, sequences, and thus it can identify and kill the cancer cell. So you've taken these T lymphocytes, they are in a dish, you've put the virus, the lentivirus goes and does transduction. These lymphocytes now start making new CAR T, chimeric antigen receptor T cell. Now that DNA in the nucleus makes RNA, RNA makes protein, protein comes out and sticks on the cell membrane. And here, then it will identify the CD19 on the cancer cell and kill it. All right. Once you have done the transduction, you have to culture, you have to multiply the cells. So I told you the dish has a culture medium, it has food for the T cells. So the transduced T cells will multiply into millions. You have to multiply because one patient needs millions of CAR T cell. We can't give one or two or five or 10. You have to grow the lymphocytes. So the cell culture creates millions of CAR T cells. This is how it looks like. It's just WBCs, right? It's like lymphocytes. And this is then given back to the doctor. And the doctor then gives these CAR T cells as an IV infusion to the patient. These CAR T cells given as an IV infusion to the patient will go inside and get attached to the cancer cell and start killing it. It's just an IV infusion of the patient's own lymphocytes. Okay, that's the beauty of it. So what we actually do is we have a patient who's not responding to chemotherapy, not responding to immunotherapy, or the cancer has come back in spite of good chemoimmunotherapy. And now they are not going to respond to anything else. At that time, we see if the cell has, say, CD19, then we talk to the genetic lab, the gene therapy lab. Hey, say, can you make CAR T cells for my patient? They say, yeah, sure. So what we actually do is we collect the patient's normal T lymphocytes by apheresis. We send this back to the genetic lab, the genetic engineering lab, where they take the lentivirus, which has this protein DNA, and they, in fact, transduct, multiply, and they increase the number of CAR T cells and send them back to me. It's like sending, you know, it's like sending 100 policemen or 100 soldiers to a military camp. In the military camp, they train them, they turn them into black cat commandos. And the 100 become 5,000. And the 5,000 black cat commandos then come back to your village to just target the terrorists, right? That is CAR T cell therapy. We infuse the CAR T cells IV, and what we actually see, this is the beauty, okay? This is the beauty of using T lymphocytes. When we infuse, suppose I'm infusing, say, 1 lakh CAR T cells or 1 million CAR T cells, okay? They go inside, they will see a lot of cancer cells. They see a lot of CD19. Now, you see the T lymphocytes can multiply. The T lymphocytes, when they identify that there is a bad cell, one T cell will become two, it will multiply. So actually what we see in 2 weeks, 7 days to 14 days is the number of CAR T cells actually increase in the blood because there are a lot of cancer cells. They multiply. One soldier makes two soldiers, makes four soldiers, right? That's the immune reaction. It's a living cell. It's a living medicine. It's a living medicine. It multiplies. And as the cancer cells die, even the CAR T cells start dying. So we don't have unnecessary CAR T cells, floating in the blood. For example, if I get COVID today, or if I get hepatitis B infection today, in the next 1 to 2 months, you will see IgM, and then it will become IgG. But if you check after 10 years, you will hardly see very little IgG, because the body's immunity is amazing. It will multiply when required and die off when not required. Yeah, that's how this is a living drug. Today, we can make CAR T cells against CD19, CD22 for B cell ALL, B cell lymphoma, and BCMA. BCMA is another protein which is found, but it is found on plasma cells. It is found on multiple myeloma. So we can make CAR T cells which can help patients with these diagnoses today. Approved therapy. Let's see what happens. You see this patient. This patient had diffuse large B cell lymphoma. This is a kind of lymphoma which has CD19 on the cell. You see, this person has axillary lymph nodes, cervical lymph nodes, retropharyngeal lymph nodes. Okay. There are mediastinal lymph nodes. Below the diaphragm, there are portal lymph nodes, mesenteric lymph nodes. All of these are cancerous lymph nodes. You give CAR T infusion, you see majority of the bad cancer cells have died. Some are still alive, but majority have died. Okay. How does this actually help? Now, this is a large study on over 400 patients these were patients who were not responding. These were B cell ALL, acute lymphoblastic leukemia, who were not responding to anything. When these patients were given CD19 CAR T cell, complete remission, 82, means in 82% of patients, the number of cancer cells went down to a level where the pathologists are not finding anything by microscope. Complete remission, 82%. Normally, we would see response in 20-30% in this group, right? Relapse refractory group. Where CAR T cell, 82% of patients, under microscope, the pathologist can't identify the cancer cells. There are such few cancer cells or no cancer cells remaining, complete remission. And even when they tried to find cancer cells by using the most sensitive technique, minimal residual disease, 69% were negative even by that. So you see, out of 100 patients, 69, there is no evidence of cancer cells. It's so effective. Normally, this will be what, 10, 20, 30%. And most of them, it stays under good control or it even cures. In some, even after CAR T cell, the cancer may come back. So when these patients are followed for 5, 6, 7, 8 years, 1/3 of them, the cancer has come back. But the other 30, 40, 45% practically cured as far as we are aware today. So these are really bad patients where the cure rate is less than 10%. Now that has gone to 30, 40, 50% or more. That is what is CAR T cell therapy. Thank you. I'll be happy to take questions.
So why isn't this tried as the first line therapy?
It will eventually become first line therapy. See, any new treatment which comes up, always, because we don't know how much it is going to help or harm, it is always used for the worst patients when they have failed everything else, right? The phase 1, phase 2, phase 3 clinical trials will always be for this. But more and more, there are clinical trials where this is used first line, because one dose and 70, 80, 90% of patients go into remission. Practically, I mean, there are of course side effects, but nothing great, nothing that can't be managed in majority. And eventually in 20 years, I believe CAR T cell will become first line and chemotherapy will become second, third line, right? Because it has side effects. But today chemo is so cheap. And even when this becomes first line, today this is very expensive. You go to America, you ask for CAR T cell therapy, you know how much it costs? It costs about 4 crores. 4 crores. In India, we have two companies which have indigenous CAR T cells, still cost about 20-25 lakhs. But 20-25 lakhs, quite a few patients can manage that. And, so eventually it will become even cheaper. All the medicines are expensive in the beginning. Eventually, this will become cheaper and, it will become first line.
Sir, I think it is a very interesting topic. First, initially, we were wondering what is this CAR T cell therapy. Now we have a very clear understanding about it. So, thank you for that simplified version. Dr. Kingsley wants to know whose lymphocyte exhibit
Autologous,
trans.
autologous.
Uh,
So, if we have 100 patients and they have blood cancer, which means they have cancer cells in the blood, when we do apheresis and we try to get their normal lymphocytes, autologous, and the genetic engineering lab will separate out the cancer cell from the. I mean, there are many more steps. It's not as easy as it sounds. So a lot of work is done by the genetic engineering lab. We give them the patients lymphocytes. They will remove it will be actually buffy coat. They'll remove the neutrophils and monocytes and cancer cells, and they'll get a concentrate of T lymphocytes, autologous, and then they will try to transduce them, and then they'll try to multiply them. So CAR T manufacture may fail in about 10% of patients. 90% of cancer today, whatever we are doing, we are able to make CAR T cells. However, there are some centers, and we are actually hoping to start a clinical trial with one such center from the US. We are in discussion, but well, we don't really know. They have created allogenic stem cell transplants, and I'm sorry, allogenic CAR T cells, which means I donate my lymphocytes, and I just donate them, right? I don't need them. I may not have blood cancer. I donate my T lymphocytes, and they transduce them, and they make ready for use allogenic T lymphocytes. So if I have a patient today, I don't have to wait for 2-3 weeks to make CAR T cell from this patient's lymphocyte. I may have ready-made allogenic T lymphocytes, CAR T cells, which can be just picked up from the shelf and transfused. So there's a lot of work going on. What the kind of CAR T cells that we are using today are only first and second generation. We are getting cells, uh, I mean, which are better, even more effective, sustain longer in the body, minimal side effects. Side effects are mainly cytokine release, etc., and all that. Yeah. So allogenic CAR T cells are also being worked on. What is also being worked on is, I said 1/3 of the patients will relapse even after CAR T. So should we do a bone marrow transplant after CAR T cell, change the complete immunity? That's another very active area of research. Yes, as of now, CAR T cell is only approved for hematologic malignancies, but there is a lot of work going on trying to manufacture CAR T cells, which will work against CNS tumors, breast cancer, colon cancer, etc. But as of now, still in research stage.
Sir, I have a small question. See, you showed the slide of a PET CT where there were multiple mets, and you said CAR T cell could be a good remedy for such cases. Such patients with advanced malignancy, we have very small time. What is the time duration required for
2 to 3. Yeah,
CAR
it
T
takes
cell
2 weeks. Yeah,
therapy
it
to
takes two, three
actually
weeks to make CAR T.
2 to 3 weeks? That is
Uh,
pretty quick considering the.
You're right. And in lymphoma, it's fine. But leukemia patients where they have cancer cells in the blood, uh,
Ah,
some of them may not survive until the point that CAR T cells are ready.
probably future maybe CAR T cell blood banks. I mean,
Yeah,
banks where
it
we
will
can.
eventually go to off the shelf. Yeah,
Yeah,
ready made. Yeah,
all
that's
right.
what is being tried. Also, this is so expensive because of the viral vector. You see, there are very few companies in the world which have US FDA viral vectors which are safe for genetic engineering. And that is very expensive. Those safe US FDA approved viral vectors are expensive. That's why CAR T is so expensive. So there is also a lot of research going on in putting in the doing gene editing without using a viral vector by using CRISPR-Cas or other enzyme systems, so that the treatment becomes cheap. That's the other thing which is happening.
Fantastic. Anybody has any other question, please unmute and ask directly, discuss with sir.
Yeah. I
Sir,
hope it is not too
Dr.
confusing.
Kingsley here.
Yeah.
Thanks for the nice explanation, and you simply simplified every complicated that CAR T cell understanding. Sir, I have a doubt that you explained the CD19 T cell receptor things. Like that, do we need to have some other receptor also for the CAR T cell?
Yes. So, for example, meningioma may not have CD19. So, any cell for which you want to make immunotherapy should have an antigenic protein which is unique to the cancer cell, which is not expressed on normal cell, and which is essential for the cancer cell. Then this will work the best, right? So it should be present on the cancer cell, essential and not. Now, CD19 is actually present on some normal B lymphocytes also. CD19 is not only on B cell malignancies, but also on normal B lymphocytes. And hence, one side effect of CD19 CAR T cell is the normal B cells also die off. And hence, the immunity may be low for a few months until the normal B cells grow back. But yes, we need, conceptually, we need some such antigen.
Thank you, sir.
You're welcome.